Hi, my name is Erica Steen, and I'm excited to share about our use of protein modeling in the interpretation of molecular consequences of missense variants in familial hemophagocytic lymphohistiocytosis, or FHLH. Perforin was the first protein to be associated with FHLH and continues to be the most frequently mutated among FHLH patients. Necessary in the lytic function of cytotoxic lymphocytes, perforin forms a pore in the target cell membrane, allowing granzymes to enter and induce apoptosis. Perforin does this by oligomerizing into a key-like pore formation conformation. Despite the frequent mutation of perforin in FHLH, many missense variants carry an unknown significance with unknown molecular consequences. Our hope was to use computational tools to further annotate and model the structure of perforin to provide insight into the potential functional consequences of missense variants. We began by first acquiring a 3D model of perforin. Although the structure of human perforin is solved, the structure predicted via AlphaFold was used for the purposes of translation of the method to proteins with unsolved structures. We then searched the literature for all experimentally determined residues with known importance with respect to the function of perforin. For example, one mutagenesis study found several residues critical for perforin's stability during its oligomerization. We then chose computational tools useful in the annotation of elements critical to both the structure and function of proteins, including conserved domains, protein-protein and ion binding sites, and solubility or solvent-accessible surface area. For features with multiple tools available, we determined a consensus and then overlaid the derived annotations onto the 3D generated structure of perforin. First, using a consensus between conserved domains database, Interpro, and available existing annotations in UniProt, we annotated the domains within perforin, including its membrane attack complex perforin superfamily domain, calcium binding domain, and epidermal growth factor-like domain. Next, PONSOL2 is a tool that substitutes every amino acid at each position in a protein sequence and provides a score for each substitution as increasing or decreasing the protein's overall solubility. We then summed all of the scores at each position in perforin sequence and created a heat map, which was overlaid onto perforin structure. This is helpful in visualizing where a missense mutation may have a more detrimental effect to the protein's structure and solubility. Next, all experimentally determined annotations from the literature, as well as those derived from computational tools, were overlaid onto the 3D structure. This included calcium binding sites, sites critical for oligomerization, residues known to be critical for perforin's lytic function, and predicted protein-protein interaction sites using the tool MetaPP-ISP. We also collected all patient-derived missense variants from ClinVar known to be either pathogenic or benign, and annotated this onto the structure. Then, for each critical residue and variant, a centroid associated with the residue's center of mass was created using Chimera X. This allowed for distance analysis between each variant and each critical residue. From this distance analysis, we found that both within the C2 and the EGF-like domains, pathogenic variants tend to cluster with predicted protein-protein interaction sites. Within the membrane attack complex domain, pathogenic variants tend to cluster with transmembrane regions and sites critical for oligomerization and lytic function. Lastly, we modeled each pathogenic and benign variant using Rosetta fold. Within Chimera X, each mutant structure was matched to the wild type, and a root mean square deviation, or RMSD value, was obtained measuring the deviation in each atom pair between the wild type and mutant structure. Shown here is the variant producing the greatest RMSD, which happens to be a pathogenic variant within the membrane attack complex domain that appears to cluster with a transmembrane region and sites critical for oligomerization. And here is colored on the structure, with red being the greatest deviation between atom pairs and blue being within 0.5 angstrom deviation. To conclude, we demonstrate the utility of computational tools in annotating proteins with the goal of providing greater insight to patient-derived variants. This approach also carries the potential to be used as a source of validation for the prediction of pathogenicity of variants with unknown significance. Thank you.